Hello, everyone, and welcome to today's keynote presentation, Paving the Path Towards Precision Medicine in Inflammatory Bowel Diseases, presented by Andrés Hurtado-Lorenzo, Senior Director of Translational Research, Crohn's and Colitis Foundation. I'm Alexis Krause of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Hurtado-Lorenzo. I will now turn the presentation over to him.
Thank you very much for your kind introduction and for the invitation to this important webinar series in precision medicine. These are my disclaimers. I have divided my presentation in 5 topics. First, I will provide a brief introduction to inflammatory bowel diseases for those in the audience not familiar with it. This will be followed by a description of current advances in precision medicine for IBD, in particular related to prognosis of disease course and response to biological therapy, followed by a description of what I believe are the biggest challenges in the field, and a description of IBD Plexus, the largest repository and research platform the Crohn's and Colitis Foundation is building to advance precision medicine in IBD. Inflammatory bowel diseases, also known as IBD, are chronic inflammatory diseases of the gastrointestinal tract. There are 2 main clinical distinct IBD presentations: Crohn's disease and ulcerative colitis. Crohn's disease is characterized by segmental inflammation that extends deep into the inner layers of the intestinal tissue, hence the term transmural inflammation. It occurs anywhere in the GI tract, and symptoms include not bloody diarrhea, weight loss, abdominal pain. In contrast, ulcerative colitis is characterized by diffuse inflammation, limited to colonic mucosa and rectum, bloody diarrhea with pus, fecal urgency, and abdominal cramps. IBD is a multifactorial pathology, and there is consensus that is the result of the interplay between genetic susceptibility, altered immune responses, intestinal microbial dysbiosis, and environmental factors yet to be determined. IBD is diagnosed using a series of laboratory and imaging tests, including routine blood and stool tests, and more specialized antimicrobial serology tests like ANCA, ASCA, SEVERE, that detect antibodies against specific microbial antigens. Imaging tests include colonoscopy and CT and MRE. Combination of these tests with histological grading of submucosal inflammatory infiltration and mucosal architecture confirms IBD. IBD has a very heterogeneous disease course, and for this reason, implementation of precision medicine practices will be ideal for targeted therapeutic management based on early prognosis. An example of this variable disease course derives from a Norwegian prospective cohort study called IBSEN that followed patients for 10 years. As shown in this figure, the disease course ranges from a disease whose symptoms severity decrease over time, shown in dark blue, to an unremitting disease characterized by that is continually active and shown in yellow. This represents 19% of patients with Crohn's disease. 32% of patients exhibiting recurrent relapsing symptoms shows in red. And a small proportion of patients, 3%, have a decrease in symptoms followed by a rampant increase in disease severity over time, shown in light blue. Overall, the cumulative disease rate in Crohn's disease was 90% during the 10 years, and 53% of patients experienced severe complications that require surgery. The main intestinal complications of IBD that require surgery and dramatically affect patient quality of life are strictures and fistulas and abscesses. According to the Montreal classification, persistent and complicated inflammation phenotype is classified as B1. Stricturing behavior is classified as B2 and is characterized by fibrostenotic narrowing of the intestinal lumen, also known as strictures. Penetrating behavior is classified as B3 and is characterized by abscesses and fistulas, which are abdominal connections of the intestine with itself and other organs, as shown in the picture. The current standard approach for treating IBD is the step-up approach that involves assessing how severe a patient's symptoms are and choosing treatment based on this severity. So, for instance, a patient who has mild symptoms probably will be started on aminosalicylates or antibiotics. A patient who has more severe symptoms or hasn't responded to the first line of treatment might be prescribed corticosteroids and immunomodulators, followed by biologics like anti-TNF alpha antibody or surgery. An alternative treatment paradigm is the top-down treatment approach. This involves treating the disease more aggressively early on and with a combination of treatments. The goal here is to induce and maintain remission. As you can see, given the complicated treatment algorithm, the implementation of precision medicine biomarkers that can stratify patients at diagnosis according to disease severity and respond to treatment will eliminate the trial and error aspects of the step-up approach or the unnecessary exposure to ineffective and costly treatment of the top-down approach. Okay, so now you begin to see what the problem is in IBD. The course of the disease is variable, with some patients having more aggressive disease than others and characterized by unremitting disease, need for drug escalation, or need for surgery. In addition, 50% of patients will experience severe stricturing complications and penetrating complications, which once they develop generally require surgery. Recurrence after surgery is frequent, dramatically impair the quality of life, and few cases might be fatal. In addition, about 30% of patients do not respond to biologics, which are the best treatment available. And they are not as primary non-responders. In addition, 15% of patients who respond lose response over time. So an urgent unmet need in IBD is the early detection, ideally a diagnosis, of patients with potential aggressive and complicated disease course, and their response to treatment, obviously. So doctors can decide the best effective treatment options based on the patient's biological and clinical characteristics to deliver the right drug at the right time, at the right dose to the right patient, and at the same time avoid the unnecessary exposure to ineffective therapies, which altogether represents the core of precision medicine concept. Precision medicine definition, defined by the NIH, is an emerging approach for disease treatment and prevention that takes into account individual variability in genes, environment, and lifestyle for each person. However, from the data I will present here, I would suggest that the definition be modified to integrate all biomolecules in addition to genes. In support of this idea, I will show you some of the cutting-edge work leading the way towards the application of precision medicine in IBD. I will show you how the integration of genotyping together with clinical and molecular phenotyping is paving the path toward prognostication of aggressive disease course characterized by unremitting disease, complications, and/or frequent relapses, and also the prognostication of the response to treatment. In particular, I will show you work related to the identification of genetic risk factors and the combination of clinical parameters with molecular phenotypes derived from transcriptomics, microbiomics, and molecular endoscopy, and their potential use as prognosis classifiers. I will start with the current clinical parameters used for prognostication in clinical practice. Current clinical features used to prognosticate disabling disease include upper gastrointestinal involvement, younger age at diagnosis, smoking, perianal disease, stricturing or penetrating disease, deep ulceration. Predictors of no response to biological therapy with TNF-alpha antagonists include the following parameters: longer disease duration, more than 2 years, small bowel involvement, smoking, normal C-reactive protein. However, these factors are not enough to accurately predict disease severity for a specific patient at diagnosis. So studies have been conducted to identify genetic risk factors of aggressive disease course and response to treatment. Currently, more than 240 risk loci have been shown to be associated with IBD. The identification of these risk loci have provided insights into the biological processes underlying the disease, such as epithelial barrier function, the effects on innate recognition and response to microbiota, as well as dysregulation of adaptive immune responses. Highlighted in red are few of these susceptibility loci that have been evaluated as potential genetic markers of prognosis. In particular, genetic variants of the NOD2 gene, which is involved in microbial recognition, has been associated with stricturing or penetrating behavior in European and North American cohorts. On the other hand, variants of the IRGM, a regulator of innate immune response and autophagy, has been associated with fistulizing disease in Italian patients. The interleukin-23 receptor, risk loci, a mediator of inflammation, has been associated with both structuring or penetrating behavior in Korean and European patients. And finally, TNF-alpha, TNF-SF15 variants have been associated with structuring or penetrating behavior in Korean patients. However, these loci still have a limited effect size that limits or restricts their use as standard markers. So in order to integrate genotyping of NO2D variants along with clinical parameters to predict disease course, Corey Siegel and colleagues created a multivariate model to predict and visualize the risk of complications. Using this model and software interface, patients and caregivers can visualize the risk, low, medium, or high, of developing complications. This model was validated in a cohort of 243 Crohn's disease patients in the PROSPECT study. This was not an inception cohort, so that already some of the patients have complications, but the exact complication data at diagnosis was available. In addition, anatomical involvement, disease location, extent of disease, and serological marker were also integrated together with this with the NO2D phenotyping. So this is an integration of clinical phenotypes together the NOD2 genotyping variants for personalized risk visualization. Now, to better characterize how genetic variation influences disease prognosis, an important large GWAS study led by James Lee and Kenneth Smith from Cambridge University in the UK identified 4 genome-wide significant loci associated with good prognosis. Importantly, none of these loci showed an association with disease susceptibility. And this is very important because at the end of the day, the main determinant of patient well-being is not diagnosis itself, but the course the disease takes over time. This study was a meta-analysis of 2 GWAS cohorts with a total of 2,734 Crohn's disease patients. Good prognosis was defined as indolent disease with no need for immunomodulators or surgery. Bad prognosis was defined as frequent flaring, treatment refractory, treatment with more than 2 immunomodulators, and more than 2 abdominal surgeries. As a result, 4 loci were associated with good prognosis, and the association were located in FOXO3, XACT, IGFBP1, and MHC. Interestingly, for example, FOXO3 expresses a protein that controls the TGF-beta1 pathway and limits inflammation, suggesting that these loci are likely involved in the biology that determines prognosis. As mentioned before, these 4 genes are not associated with susceptibility, which suggests that disease initiation is governed by different genetics to that drives the disease course. And this is a very important aspect that suggests that the biology underlying prognosis not only have a prognostic predictive value, but opens the doors for novel therapeutic approaches to control disease progression. So due to the limited effect size of single loci and as a standalone classifiers, the incorporation of several polymorphisms and generation of genetic risk scores have been proposed as better approach to increase predictive value of these genetic classifiers. As example of this is a very recent work by the group of Ramnik Xavier at MGH, recently published in the official journal of the Crohn's and Colitis Foundation, Inflammatory Bowel Diseases. The goal of this study was to generate genetic risk score to predict primary no response to anti-TNF alpha antibody and duration of response in 231 patients with ulcerative colitis. Patients were genotyped using Illumina immunochip, and primary no response was defined as no response at week 12 of treatment initiation. Durable response was defined as maintenance of response 20 months after 20 months of treatment. Response was evaluated using, in addition to the genetic information, several clinical parameters that were evaluated. So the outcomes were the following. As shown in the left panel, 7 SNPs were associated with primary no response. Importantly, when clinical parameters were used alone to predict primary no response, the RC was only 0.6. In contrast, the genetic risk score based on these 7 SNPs improved the predictive value for primary no response with an ROC of 0.85. And combined genetic and clinical models was very similar to the genetic risk score alone. Now, regarding the prediction of durable response, 12 SNPs were associated with durable response. And similar to the prediction of primary no response, the genetic risk score alone was superior than the clinical model to predict durable response with an RC of 0.78. Now, a combination of clinical and genetic model, once again, did not improve the genetic risk score, while very promising, and what this is very promising, larger prospective studies are required to confirm clinical utility of these genetic risk scores. In summary, I I have shown you that genotyping has been explored as prognostication tool to predict aggressive disease course and response to treatment. Several loci associated with IBD prognosis have been identified, some of which are also susceptibility loci, and others are purely prognosis loci, suggesting that the contribution of genetics to disease progression is independent from the contribution of genetics to disease susceptibility. However, their clinical implication or application still is limited. And finally, some data has been generated that supports the notion that composite risk score might provide an improved prognosis value and effect size than using single loci as standalone classifiers. Okay, now we'll shift gears to highlight very exciting advances in the integration of clinical parameters along with molecular phenotyping using transcriptomics for IBD prognosis. I will first refer to a groundbreaking work related to the prognosis of a complicated disease course and response to treatment in a pediatric IBD cohort known as the RISK study. This cohort is the largest well-characterized inception cohort in pediatric IBD, in which patients were enrolled at diagnosis and therefore before treatment. The aims of this study were to identify clinical and biological predictors of complications in Crohn's disease at diagnosis, and to evaluate the effect of early anti-TNF alpha treatment in reducing the risk of complications. The RISK pediatric inception cohort started in 2008. It is funded entirely by the Crohn's and Colitis Foundation. It consists of 28 sites, 25 in USA and 3 in Canada. 1,800 pediatric patients were enrolled, of which 1,000 are Crohn's disease patients. Clinical data and biosamples were collected, and information was collected every 6 months for 5 years. The 3 years follow-up outcomes were published in a seminal paper in Lancet by principal investigators Subra Kugathasan and Lee Delson and collaborators. The methodology consisted of data collection related to clinical features, antimicrobial serologies, gene expression, microbiome, genotyping, and response to treatment. Computational and biostatistical tools were used to analyze 80 million data points in order to generate a competing risk model for prediction of complication and a propensity score matching to assess early anti-TNF alpha efficacy. The outcomes of the 3 year follow-up were the following. According to the Montreal classification, out of 913 patients that remained, 835 remain inflammatory or B1. 54 children developed structuring complications, B2, and 24 children developed penetrating complications, or B3. As shown in this survival curve, 10.7% of patients develop complications by GR3, either penetrating or structuring complications. Now, the base prediction model using clinical phenotype showed that severe positive serology that measures antiflagellant antibodies was associated with structuring behavior, and ASCA positive, severe positive, older age at diagnosis, and African American race were associated with penetrating complications. To identify potential prognosis predictors based on gene expression profiling, ileal biopsies obtained at baseline colonoscopy were used to isolate RNA and to evaluate global gene expression profiles using RNA-seq. These are very exciting results showing that prognostic gene expression signatures were identified. These figures show that those patients who develop penetrating complications like internal fistula and/or abscesses by year 3 exhibited at diagnosis upregulating expression of genes involved in inflammatory processes. And these children developed a complication which is inflammatory in nature, like fistulas and abscesses. In contrast, children who at diagnosis had increased expression of extracellular matrix genes, ECM, went to develop structuring complications, which are characterized by fibrostenosis due to mucosal and submucosal deposition of extracellular proteins, extracellular matrix proteins. These signals are therefore consistent with the biology of the complication. And this suggests that the gene expression signatures might not only be potential prognosis biomarkers, but drivers of the biology of complications. And therefore, they can be potential targets for therapeutic discovery, a concept which is consistent with that of the GWAS prognosis loci that I mentioned before. This slide shows that the composite risk model, based on clinical features, had an outlook of 0.66. An integration of the ECM gene signature increased the outlook to 0.72 and the specificity to 71%. The positive predicted value also increased, and the negative predicted value remained very high with or without gene expression. Now, to evaluate the effect of early therapy with anti-TNF alpha in reducing risk of complications, children were treated with this biologic drug within 90 days of diagnosis, which is defined as early treatment, and analyzed via propensity score matching. The complication-free survival curve on the right shows that early treatment with anti-inflammatory antibody reduced the risk, anti-TNF alpha antibody reduced the risk of penetrating complications shown in red, but did not reduce the risk of developing structuring complications shown in blue, which is consistent with the notion that an inflammation-independent fibrotic process drives the biology of the structuring complication, as might be predicted from the regulated profibrotic signature this patient exhibits early in the disease. All right, so in summary, RISK is the largest study of newly diagnosed pediatric Crohn's disease patients. A risk prediction model for complicated Crohn's disease at diagnosis based on clinical and gene expression data was generated. In this model, extracellular matrix gene signatures is associated with structuring disease, and inflammatory gene signatures is associated with penetrating disease. Also, early anti-TNF alpha therapy reduced penetrating but not structuring complications. So finally, this competing risk model that includes clinical and gene expression data could help stratify patients to decide the best personalized treatment options at diagnosis. A subsequent paper from the RISK consortium, from the same cohort, the same study, evaluated the possibility of using transcriptional risk score to predict complicated cures. For this, published GWAS and EQTL's data were integrated with RNA-seq studies from RISK. This led to the identification of 29 genes able to distinguish disease status and, more importantly, prediction of structuring and penetrating complications, as you can see in the right-hand panel. More importantly yet, the transcriptional risk score was superior to predict complications compared to the genetic risk score. So it's a very new concept and very promising concept for prognosis. Okay, so I show you very important evidence to support the use of gene expression signatures and transcriptional risk scores to predict complications in IBD, like structuring or penetrating complications, and predict response to treatment. But what about prediction of aggressive disease course, characterized by frequent relapse and the need for treatment escalation? Okay, in this regard, this is another groundbreaking study from the lab of Kenneth Smith in Cambridge, UK, that aimed to identify gene expression signatures for prediction of aggressive disease course. In this study, aggressive disease was defined as unremitting disease, which is continuously active and with frequent relapses, and that requires therapy escalation from immunomodulators all the way to surgery. In contrast, quiescent disease was defined as sustained remission in which did not require therapy escalation. For the study, 35 patients with active Crohn's disease and 32 patients with active ulcerative colitis were enrolled. 58% of patients were enrolled at diagnosis. PBMCs were used to isolate CD8 T cells and CD4 T cells, and their transcriptional profile analyzed using Affymetric arrays and qPCR. As mentioned, these patients were managed with a step-up approach. As shown here, transcriptional profiling of CD8 T cells from patients resulted in the identification of two gene expression signatures called IBD1 and IBD2 that stratify Crohn's disease and ulcerative colitis patients in two well-defined groups. Namely, those with the IBD1 signature are characterized by elevated expression of differentially expressed genes, and those with IBD2 signatures in blue have lower expression of differentially expressed genes. More importantly, the ability of these gene expression classifiers to predict disease course both in Crohn's disease and ulcerative colitis was also evaluated. In particular, those patients expressing the IBD1 signature in red had more aggressive disease course than that required drug escalation. In contrast, those patients expressing the IBD2 signature in blue exhibited an indolent disease which did not require drug escalation. So importantly, the use of these gene expression classifiers was significantly superior to that of a clinical risk score or antimicrobial serologies with ASCA to predict disease course, as shown in the middle and right panels. These gene expression signature-based classifiers represent a very important advance in IBD precision medicine, as it will allow the decision at diagnosis of whether to implement a top-down therapeutic approach in which patients with aggressive prognosis will be treated early with biologics, and on the other hand, it will prevent the unnecessary exposure to the side effects of biological immunosuppression in patients with indolent disease. The utility of these gene expression signatures to guide the treatment choices between a standard step-up therapy and the first-line use of biological immunomodulatory drugs is currently being evaluated in a randomized controlled trial called the Profile study. Okay, so I provided examples of gene expression signatures to prognosticate structuring and penetrating complications, as well as aggressive disease course characterized by frequent relapse and the need for treatment escalation. So what about prediction of response to treatment, in particular biological therapy using anti-TNF alpha or anti-integrin blockers, the most common biologics used in IBD? A very important work in this regard comes from the lab of Fiona Puri from Oxford University in the UK. They analyzed a number of public gene expression data sets and identified 65 differentially abundant cytokines in IBD compared to non-IBD controls. Importantly, among all these cytokines, it was the oncostatin M cytokine gene, the highest expressed one in Crohn's disease patients. More importantly, they demonstrated that increase in oncostatin M expression correlates with disease severity, as shown in the right panel, which was defined by histological score. And this means that the higher the levels of oncostatin M are, the more severe the disease is. To answer the question of whether oncostatin M could be a predictor of response to biological therapy, they analyzed several observation and prospective cohorts. As shown in the left panel, 90% of patients expressing the so-called high expression of oncostatin M module in red were refractory to anti-TNF alpha antibody therapy as expected. In contrast, 85% of patients with low expression of the oncostatin M module were responsive to anti-TNF alpha therapy. The endpoint to assess response to therapy treatment was mucosal healing. Another analysis using the two prospective cohorts from a phase 3 clinical trial for TNF alpha antibody, it was found that before initiation of the treatment, oncostatin M levels were already higher in those patients with refractory ulcerative colitis compared to those patients who achieved remission or had partial response. Importantly, the expression levels of oncostatin M remained high 8 weeks after treatment initiation in those patients that did not respond to treatment. So in summary, I have showed you several examples of very important advances in the identification of gene expression signatures that could be used for early prognosis of complications using the ECM and in the inflammatory signatures from the risk study. In addition, other signatures like the IBD1 and IBD2 can stratify patients who will develop an aggressive disease or an indolent disease course, and other signatures like the oncostatin M and the ECM to predict response to treatment. Additional prospective randomized controlled trials will help determine whether these transcriptional classifiers could indeed be used to decide the best treatment options early in the disease and improve patient outcomes. Okay, now I will switch gears again to present experimental evidence supporting the integration of clinical phenotyping with microbiomics analysis for prognosis of complicated disease course and response to treatment. The risk study that I mentioned earlier did not only focus on the study of gene expression signatures, but also evaluated changes in gut microbiome as potential predictors of complications in pediatric Crohn's disease. As shown in the left panel, 14 genera were associated with pediatric Crohn's disease, 4 of which were increased, in particular Agrigobacter and Campylobacter. In contrast, 10 other genera were decreased in Crohn's disease. More importantly, as shown in the right panel, 2 taxa, Ruminococcus and Rothia, were associated with development of stricturing disease. And Colnicella and Bellionella are associated with fistulizing disease. These results are currently being evaluated for inclusion in the risk composite model I described earlier. All right. Another very important study derived from the Crohn's and Colitis Foundation Microbiome Initiative is related to the investigation of microbiome-based predictors of response to anti-integrin antibody and other biologic drug used for the treatment of IBD. For these 42 Crohn's disease patients and 43 ulcerative colitis patients from the PRINCEM cohort at MGH and initiating anti-integrin therapy were included in the study. The primary outcome was defined as clinical remission at week 14. Remission was evaluated by reduction on the HBI and SSCA disease scores. Microbiome studies were done using stool samples and analyzed with shotgun metagenomics. So what were the outcomes? 31 patients achieved remission by week 14, 35% retained remission, and the clinical features associated with remission were shorter duration of disease, most likely Crohn's disease diagnosis, and less likely anti-TNF alpha exposure, the other biological agent used in IBD treatment. Regarding the microbiome analysis before treatment, there is an increased abundance of only 2 taxa associated with patients who responded to treatment and achieved remission by week 14. In contrast, when microbial function was evaluated, 13 pathways were upregulated in responders. At week 14, abundance of phytaxa were decreased, including Prosperia in eluborans that is increased before treatment, as you see in the upper panel. They also found a decrease in 17 biological pathways, including most of those upregulated at baseline before treatment. Now, importantly, they implemented a neural network analysis to integrate and train the system to incorporate clinical parameters, microbial taxa abundance, and microbial pathways. This led to an increased predicted value of response to treatment with an IUC of 0.87. Clinical parameters alone had only an IUC of 0.69. So these important results not only support the use of microbial signatures as prognostic markers, but how the use of artificial intelligence can help to make sense of all this information coming from the omics analysis. Finally, I will highlight current efforts in the area of molecular endoscopy for prediction of response to treatment. As most of you know, anti-TNF alpha antibodies used in IBD therapy work by binding both to soluble and membrane-bound TNF alpha. In this study, the hypothesis was that quantification of the levels of membrane-bound TNF alpha in the patient's mucosa can identify responders to TNF alpha therapy. To test this hypothesis, FITC-labeled anti-TNF alpha antibody, also known as adalimumab, was delivered topically via a spray catheter. Confocal laser endoscopy was used to visualize cells with membrane-bound TNF alpha. The study was made in 25 Crohn's disease patients with active disease, defined as Crohn's disease activity index bigger than 150. Molecular endoscopy was done before treatment, and this was day 0. Adalimumab treatment started 7 days later, and response was measured by reduction in Crohn's disease activity index at day 31 and 91. No response was defined as need for surgery. As shown in the right panel, as predicted, patients with the high levels of membrane-bound TNF alpha responded to anti-TNF alpha therapy. Patients with low levels of TNF alpha in the membrane did not respond to therapy. Overall, 96% of patients with high levels of TNF alpha in the membrane respond to treatment. In contrast, only 15% of patients with low levels of TNF alpha responded to treatment. The assay was highly sensitive and specific, and very high positive and negative predictive values. And a more recent study published by the same group also found that patients with high levels of integrin alpha 4 beta 7 in the membrane were better responders to anti-integrin therapy with vedolizumab compared to those with low levels of membrane-bound integrin. All right, so in summary, I highlighted very exciting progress made in the identification of gene expression, microbiome, and endoscopic prognostic signatures that could be useful in predicting complications like stricture and penetrating, aggressive disease course characterized by chronically active disease with frequent relapse and need for treatment escalation. And these molecular and microbial signatures may also predict response to treatment. Okay, so given all this experimental evidence, it is clear that clinical phenotyping is just the tip of the iceberg in prognosis and personalized IBD treatment. And therefore, deep phenotyping of patients at the molecular level is needed to advance the concept of precision medicine in IBD. It also appears that multi-omics data integration seems to be key for generation of prognostic classifiers with improved predictive value for patient stratification in order to decide efficacious personalized treatments early in the disease. Okay, so now despite these advances, several challenges still lay ahead, in particular related to data collection and integration. There is a big need for harmonized data and sample collection for subsequent multiomics data generation and integration. Challenges are also in the integration of electronic health records with multiomics data and the need to develop artificial intelligence tools to support the integration and analysis. A very important need is the validation of candidate prognosis biomarkers in longitudinal prospective cohorts and inception cohorts, which are ethnically diverse. And even more important, assessment of the clinical utility of these markers in rigorous randomized controlled trials to demonstrate improvement in patient outcomes and cost-effective care. As an approach to circumvent these challenges, the Crohn's and Colitis Foundation is building the largest repository of IBD biosamples and linked patients' data, in which harmonized protocols and methods are used to enroll patients and collect samples. These samples will be used for harmonized generation of multiomic profiles that can be integrated with electronic health records. IBD Plexus is therefore a research platform that provides a specialized biobank infrastructure, analytical platforms for data mining, central reference labs where samples will be converted into molecular data using harmonized protocols with the highest industry standards. And finally, Plexus also confers an infrastructure with high performance computing and research portal for access data. IBD Plexus consists of 4 research cohorts, 2 virtual cohorts that collect patient-reported outcomes and data and quality of care data. They are called IBD Chorus and IBD Partners. And there are about 2 additional cohorts, an adult prospective cohort called a Spark, and Risk, the Inception cohort I described earlier. Both collect clinical data and biosamples. In particular, a Spark is a prospective adult cohort that aims to enroll 7,000 patients by the end of 2020. Clinical data and biosamples will be collected. Risk is the legacy pediatric Inception cohort that conceived and funded by the Crohn's and Colitis Foundations for the last 10 years. This cohort provided the groundbreaking work on prediction of complications described earlier, and about 1,000 pediatric patients were enrolled and biosamples were also collected. So in conclusion, I showed you today, and I hope I convinced you that very exciting advances have been made in the last decade in regard to integrating genotyping and deep clinical and molecular phenotyping for IBD prognosis. In particular, the prognosis of a structuring and penetrating complication at diagnosis, the prognosis of aggressive disease course characterized by persistent relapses and the need for drug escalation, and finally, prediction of response to biological therapy. Altogether, these prognostic classifiers might help doctors decide at diagnosis and with precision in which patients to implement a step up or a top-down approach for effective treatment. These studies also are opening the doors to identify new targets for drug discovery of therapies tailored to the biological features of these patients. Together, this research and future research, including research derived from the IBD Plexus platform that the Crohn's and Colitis Foundation is building, is paving the path towards precision medicine in IBD. However, more work is still required for its clinical implementation and change in patient outcomes. Thank you very much, and I'm very glad to take questions.
Thank you, Dr. Hurtado-Lorenzo, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window, type your question into the box that appears on the screen, and click the Send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, you presented data related to the use of molecular markers for prediction of disease course in response to treatment. Has any work been done regarding the prediction of side effects of current IBD treatments?
Yeah, thank you for that. Actually, that's a very good question. And there are at least 3 examples of genetic variants that are associated with toxicity to thiopurines, a type of drug that is commonly used to treat IBD. First, it's well established that genetic polymorphisms in the thiopurine S-methyltransferase, which is also known as TPMT, leads to reduced TPMT activity, and this results in the accumulation of a toxic metabolite of thiopurines, which is associated with development of myelotoxicity. Another polymorphism that increases the toxicity of thiopurines is the NUDIX hydrolase, also known as NUDIT15 gene. This SNP is associated with leukopenia in patients exposed to thiopurines. And a third polymorphism is the class 2 HLA gene region that has been associated with pancreatitis in patients exposed to thiopurines. And we predict that there will be more work in the identification of polymorphisms linked to the side effects of biological treatment as well.
Our next question is, are environmental factors being studied for prediction of disease course?
Yeah, thank you for that question. This is a very timely question. As I mentioned in my presentation, smoking, which is one of the most studied environmental factors in IBD, is related with bad prognosis of Crohn's disease. However, it's important to understand the role of other environmental factors in disease susceptibility and prognosis. With that aim in mind, the Crohn's and Colitis Foundation recently launched an initiative to identify biological signatures of disease susceptibility and progression, and in response to environmental exposures. In particular, these studies are currently evaluating the identification of microbial signatures, brain imaging signatures, and vascular signatures to predict frequent disease relapse in response to exposure to psychological stress, either daily perceived stress or early life stress. We're also searching for antiviral antibody signatures in response to hit-and-run virus exposures that can trigger disease relapse. If positive, a simple blood test analyzed using viral multipeptide arrays might predict the development of frequent relapse course in response to viral exposures. So, and finally, the pediatric risk cohort that I mentioned are also evaluating the epigenetic signatures to evaluate the role as predictors of complications. So in conclusion, the exposome field is growing in IBD, and I'm sure that it will make part of the armamentarium of measurements that will support effective precision medicine in IBD.
I would like to once again thank Dr. Hurtado-Lorenzo for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through September of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.